All right, welcome back to the deep dive. Today we're diving into a paper that caught my eye, multimodal language modeling for high accuracy single-cell transcriptomics analysis and generation.
Wow, that's a mouthful.
Yeah, it is.
Definitely a mouthful, but a really fascinating paper. And I think, the title kind of gives us a clue as to what we're going to be talking about.
Yeah, it sounds complex, but at its heart, it's all about using a new machine learning model, this SCM-GPT thing, to get a much deeper understanding of what's going on inside individual cells.
Right, and it does that by, connecting the dots between all that raw genetic data we have from single-cell sequencing and the vast amount of knowledge we've accumulated about cells from scientific papers, textbooks, even Wikipedia.
And that connection, that's what really makes this paper interesting. I mean, we've all seen those heat maps with thousands of genes and their expression levels in different cells. It's a lot of data, but on its own, it can be really hard to make sense of.
It's like, imagine trying to understand a city just by looking at a map of all the buildings and streets, but with no labels or any information about what those buildings actually are or what people do inside them.
Yeah, you'd have no idea if you were looking at a hospital, a school, or a giant shopping mall. You need that extra layer of context.
Exactly. And that's what SCM-GPT brings to the table. It's like adding all those labels and descriptions to our cellular map. It learns to read both the genetic code and human language, so it can point to a specific pattern of gene activity and say, hey, this looks like a neuron that's involved in memory formation.
So it's kind of like a Rosetta Stone for cells, helping us decode those complex genetic signals.
That's a great analogy. It's essentially bridging the gap between these two very different but incredibly important sources of information, the genes themselves, and the vast body of scientific knowledge about what those genes actually do.
So before we jump into the nitty-gritty of how this model works, let's take a quick step back and look at the history that led to this research.
Okay, so where do we start?
Well, I think we need to talk about the huge impact of pre-trained language models, or PLMs, in science in general. I mean, we've all heard about how models like GPT are revolutionizing natural language processing, allowing computers to understand and generate human-like text.
Yeah, and it was only a matter of time before people started exploring how to apply these powerful techniques to biological data, especially with the explosion of data we're getting from single-cell sequencing.
Right. Early efforts focused on using text-based PLMs to do things like automatically labeling different types of cells based on text descriptions or, retrieving information about cells using text queries, kind of like a supercharged search engine for cell biology.
And then came the cell PLMs, which were specifically designed to understand the patterns in gene expression data from cRNA-seq.
Yeah, these models treated gene expression profiles like sentences, where the genes were the words. And they've actually been really successful in things like correcting for technical differences between experiments, which is a huge problem in single-cell analysis, or even generating artificial cells, those pseudocells you mentioned earlier.
Right, but there was a big problem. You had these text-based models that were great at understanding text, but they couldn't make heads or tails of the raw gene expression data.
And you had these cell-based models that were great at understanding gene expression, but couldn't process any free-form text.
Exactly. It was like having two sets of experts who spoke completely different languages and couldn't collaborate effectively.
And some earlier attempts to bridge this gap, well, they weren't perfect.
Yeah, one approach that was pretty popular was this idea of creating cell sentences. Basically, they'd take a cell's gene expression profile, pick out the top, say, 30 or 100 most highly expressed genes, and string their names together into a sentence.
So instead of having this rich, multidimensional data about thousands of genes, you just have this short, oversimplified sentence.
Right. And as you can imagine, a lot of crucial information gets lost in that translation. It's like trying to describe an entire movie just by listing the names of the main characters.
You'd miss all the nuances of the plot, the relationships, the whole point of the story.
Exactly. And figure 2 in the paper really drives this point home. They show that using these cell sentences as input led to a significant drop in accuracy when it came to identifying different cell types.
And another issue they highlight is the problem of insufficient pre-training across both data types. Some early attempts to use text models for cell analysis just didn't train them on enough single-cell data for them to truly grasp the complexity of cellular states.
And on the flip side, sometimes those large language models would be trained on cellular data, but they would kind of forget some of their core text processing abilities in the process. It's like, a brilliant chef who tries to learn a completely new cuisine, might end up forgetting some of their basic culinary skills.
So this is where SCM-GPT comes in, right?
Yep, exactly. And it's designed to overcome these limitations by creating a unified language model that can understand and generate both cell and text data without losing critical information along the way.
So how does it do that?
Well, they start with two powerful existing models. SCGPT, which is a well-established cell PLM that's really good at understanding complete CRNA-seq data, and Llama 2, which is this very capable text-based language model.
So those are the two experts who speak different languages.
Yeah, and the key to getting them to work together is through what they call cross-modal projectors.
Cross-modal projectors.
Yeah, think of them as sophisticated translators that can take the language of gene expression, those complex patterns of up and down regulation, and translate it into a format that the text model can understand.
And vice versa.
Exactly. And they actually use two types of projectors. A cell-to-text projector, which they call a Q-former, and it's initially informed by knowledge from this other language model called BioMedBERT that focuses on biomedical text, and a text-to-cell projector, which uses this technique called cross-attention.
So the cell-to-text projector helps the model generate descriptions of cells based on their gene expression, and the text-to-cell projector helps it generate those artificial pseudocells based on textual instructions.
That's it. It's pretty clever how they put it all together. And then to really make this model shine, they trained it on a massive data set of 27 million single cells from the CellX gene database.
Wow, 27 million, that's huge.
It is. And as Figure 4 shows, this dataset covers a really diverse range of human organs and tissues, which is really important for making sure the model can generalize its knowledge to different types of cells. In fact, it's the largest dataset anyone has ever used to train a model that can understand both cell and text data simultaneously.
And it seems like all that hard work paid off. I mean, the results they present are pretty impressive.
Yeah. For example, when they tested SCM-MGPT's ability to generate text descriptions of cells based on their gene expression, they found an 84% relative improvement in what they call textual discrepancy.
Textual discrepancy.
Yeah, it's basically a measure of how different the generated descriptions are from the actual real-world descriptions of those cells. The lower the discrepancy, the better the model is at capturing the essence of those cells in words. And a reduction of 84% is like a massive leap forward.
Okay, so the model is getting really good at describing cells based on their genes. What about the other tasks, like identifying different cell types?
Oh, there. SCM-MGPT achieved a 20.5% higher accuracy compared to existing methods.
That's a pretty significant jump, isn't it?
Yeah, it definitely is. And for generating those synthetic pseudocells based on text descriptions, they saw a 4% improvement. Now, that might not sound like a lot, but in this field, even small improvements can be a big deal.
Okay, so the results are clearly very promising. Now, let's dive a little deeper into the paper and see how they got there. What did they do in their related works section?
So in the related works section, they basically walk us through the history of the field and lay out the foundation for their own work.
Okay.
They highlight, all the progress that's been made in single-cell sequencing technologies and the development of those early cell-focused language models we talked about.
Okay.
They also do a good job of reviewing those earlier attempts to connect cell data with text, like the cell sentence approach. And they very clearly point out the limitations of those methods.
Right, which sets the stage for why their approach using SCM-MGPT is such a big deal.
Exactly. They also draw parallels to the success of similar multimodal language models in other scientific areas, like understanding small molecules and proteins, which gives their approach even more credibility.
So how about the methods section? What kind of details do they provide there?
So in the methods section, they really get into the nitty-gritty of how SCM-MGPT is built and how it's trained.
Makes sense. That's where the technical magic happens.
Right. And for those of you who are interested in the technical details, they start by describing all the data pre-processing steps they use for the cRNA-seq data, like normalization and log transformation, which you can see in equation 1.
Okay, that's pretty standard stuff for anyone working with gene expression data.
Yeah, it is. But then they go into how they collected and processed all that text data from Celtix gene, the OBO Foundry, which is this collection of biological ontologies, and even Wikipedia.
Ah, so they're pulling information from all these different sources to give the model as much context as possible.
Exactly. They break down all that text into smaller units called tokens, which are like the individual words or pieces of words that the model learns from.
Okay.
And then they describe in more detail those two core components of the model, SCGPT for understanding the cell data and Llama 2 for processing the text. They explain why they chose these particular models and how they've been pre-trained on massive amounts of data to give them a solid foundation.
Makes sense. Start with the best possible building blocks.
Right. And then the real star of the show, as we've discussed, are those cross-modal projectors.
Those are the translators, right?
Yeah, exactly. The cell-to-text projector, that Q-former thing, is particularly interesting.
How so?
Well, it uses these learnable query tokens, think of them as little probes, that interact with the output of the cell model, SCM-GPT, to figure out which parts of the complex gene expression data are the most relevant for generating a text description.
So it's not just blindly passing all the data to the text model, it's selectively picking out the most important pieces.
Exactly. And by initializing this projector with knowledge from BioMedBird, they're essentially giving it a head start in understanding biological concepts.
Makes sense. Kind of like having a translator who already knows a lot of the specialized vocabulary.
Yeah, that's a good way to put it. And then the text-to-cell projector uses cross-attention, which allows the text information to directly influence the cell model's understanding, guiding it in the process of generating those pseudocells.
So it's like giving the cell model a set of instructions or a soft prompt in the form of text.
Exactly. It's like saying, okay, cell model, I want you to create a pseudocell that has these specific characteristics. And the model can use that textual guidance to figure out what kind of gene expression profile would be needed to create that kind of cell.
So how do they actually train this whole thing? How do they teach it to understand both cell and text data and make these connections?
Well, they use a pretty sophisticated training process that involves two main objectives.
Objectives?
Yeah, think of them as the goals that the model is trying to achieve during training. The first objective is what they call cell text representation alignment.
Okay, so that's where the model learns to recognize which cells and text descriptions belong together.
Exactly. And they use two different loss functions to do this. A cell text contrastive loss, which is shown in equation 2, and a cell text matching loss in equation 3. These loss functions are just mathematical ways of telling the model when it's getting it right or wrong during the training process.
Right, so the model is constantly being adjusted to make those connections between cell data and text descriptions more accurate.
Exactly. And the overall alignment loss, which they call L-align and show in equation 4, combines those two individual losses to guide this part of the training.
Okay, and the second objective?
The second objective is cell text translation, where they focus on the model's ability to generate text descriptions from cell data and vice versa.
So that's where they use the cell description generation loss, equation 5, and the pseudocell generation loss, equation 6, right?
Yep, those are the ones, they combine those into an overall generation loss, LGen, as shown in equation 7.
Okay, and they mentioned a two-stage pre-training approach. What's that about?
Yeah, so in the first stage, they focus on getting that fundamental link between the cell and text representations established by training the cell model and the cell-to-text projector using that LA line loss we talked about.
So that's like the initial alignment phase where the model learns to recognize those basic connections.
Right. And at this point, the model can already be used for tasks like cell type annotation, even without the full text generation capabilities.
Interesting.
And then in the second stage, they freeze those first stage components and focus on training the text to cell projector and fine-tuning the text model using the LGen loss.
So that's where the model really learns to generate those text descriptions and pseudocells.
Exactly. By separating the training into these two stages, they can really optimize each part of the model for its specific task.
Okay, makes sense. Now, what about the result of all these experiments? How did SCM-GPT actually perform?
So as we mentioned earlier, the results are really promising. For cell description generation, they show in Table 1 significant improvements in both MMD and EMD scores, which are measures of how similar the generated descriptions are to the real-world descriptions.
And those improvements were backed up by statistical tests, right?
Yeah, the large reductions in MMD and EMD scores, along with those statistically significant p-values from the t-test and ks test, really give us confidence that the model is doing something truly remarkable.
So it's not just a fluke. The results are statistically robust.
Exactly. And for generating those text-guided pseudocells, as shown in Table 2, SCM-GPT achieved state-of-the-art performance across various ways of measuring the similarity of the generated cells to real cells.
So again, statistically significant improvements over existing methods.
Yep, and this really suggests that the model can create biologically relevant synthetic cells based on those textual instructions, which has huge implications for research and potentially even for developing new therapies.
Yeah, I can see how that would be useful. Now, how about the cell type annotation results? Did they use that Tabula sapiens dataset?
They did, yeah. And this is where it gets really interesting because that data set contains a lot of cell types that the model had never seen during its initial training.
So it's like testing the model on a completely new language.
Kind of, yeah. And the fact that SCM-GPT still performs so well in the zero-shot setting, as you can see in Table 3, really highlights how well it can generalize its knowledge to new cell types.
So it's not just memorizing the cells it saw during training, it's actually learning something fundamental about how gene expression relates to cell identity.
Exactly. And when they fine-tuned the model on that data set, it showed even further improvements, which is really encouraging.
Okay, so they have all these great results. Did they do any ablation studies to understand what parts of the model are really driving those improvements?
Yes, they did. In Table 4, they compare the full CM-GPT model to a version that only uses text, and the results are pretty clear.
What did they find?
Well, they found that the cell understanding component, SCGPT, is absolutely crucial for generating those high-quality cell descriptions. When they remove that component, the performance dropped significantly.
So that really highlights the importance of having that dedicated cell-focused language model as part of the system.
Exactly. And they also experimented with different sizes of the underlying text model, using Tiny Llama and Llama 2 to see how the text model's capacity affects performance.
What did they find there?
Well, they found that using a larger text model, like Llama 2, does generally lead to better results. But even with the smaller Tiny Llama model, SCM-GPT still outperformed those baseline methods that didn't use any cell-specific language modeling.
Okay, so it seems like both the cell and text models play important roles, but the cell model, SCGPT, seems particularly crucial.
Yeah, that seems to be the case. Now, the visualization section also offers some really helpful insights.
Visualizations are always nice for getting a more intuitive feel for what the model is doing.
Definitely.
Yeah.
In Figure 2, they visually demonstrate how using those limited cell sentences as input leads to a big loss of accuracy in cell type identification.
So that's another nail in the coffin for that approach.
Yeah, pretty much. And in Figure 6, they show that using more genes to represent a cell moving beyond just those top 30 or 100 leads to better performance in cell description generation.
So more information is generally better, which makes sense. What about Figure 5?
Figure 5 is a really cool visualization that shows how SCM-GPT can effectively remove those technical batch effects from the data while still preserving the biological differences between different cell types in its learned representations.
Batch effects.
Yeah, those are technical variations that can arise between different experiments, and they can really mess up your analysis if you're not careful.
Right, because you want to make sure you're comparing apples to apples, not apples to oranges that have been contaminated with, like, grapefruit juice.
Exactly. And SCM-GPT seems to be really good at separating the true biological signal from the noise introduced by these technical variations.
That's a really valuable capability for anyone working with real-world single-cell data.
Absolutely. So, in their conclusion, the authors reiterate that SCM-GPT represents a significant leap forward in single-cell analysis by successfully integrating cRNA, seq, data, and text for a wide range of tasks.
And they really emphasize the importance of those cross-modal projectors in making that integration possible.
Yeah, those projectors are the key innovation that allows the model to bridge the gap between those two worlds of data.
And of course, they highlight the superior performance they've achieved thanks to all that large-scale pre-training.
Right, training on that massive data set of 27 million cells was really crucial for getting these kinds of results.
And they also point to some exciting future directions for this work.
Yeah, they talk about expanding the model to understand data from more species beyond just humans, which would be really valuable for researchers studying model organisms like mice or even plants.
Right, because right now their training data is heavily focused on human cells, which might limit its applicability to other organisms.
Yeah, that's one of the limitations they acknowledge.
What are some of the other limitations?
Well, they also point out that they haven't yet integrated other types of single-cell data beyond just transcriptomics, like SCATAC-seq, which measures DNA accessibility, or CIT-seq, which measures protein levels.
So right now they're really focused on the RNA abundance part of the story, but there's so much more to a cell than just its RNA.
Exactly. And being able to integrate those other layers of information would definitely make the model even more powerful.
They also mentioned the potential for integrating different types of omics data, like genomics, proteomics, metabolomics, and even translating between them. That would be really amazing.
It would be like having a universal translator for all these different biological data sets.
And they even suggest that these kinds of models could eventually help us discover entirely new cell types that we didn't even know existed.
That's a really exciting possibility.
Yeah.
I mean, we're still just scratching the surface of understanding the incredible diversity of cells that make up our bodies and all other living organisms.
So for our listeners who are interested in machine learning and its applications in biology, SCM-GPT represents a really important step forward. It shows that by combining different types of data, in this case, gene expression and text, and using these sophisticated language modeling techniques, we can really unlock new levels of understanding in single-cell biology.
Absolutely. And this could lead to all sorts of amazing breakthroughs like more accurate cell identification, better understanding of how diseases work at the cellular level, and even the development of entirely new therapies.
And for those who want to delve deeper into the technical details, we've got a link to the full paper in the show notes. Definitely worth a read if you're interested in this kind of cutting-edge research.
Now, before we wrap up, I think it'd be fun to do our usual novel notions segment where we take a moment to brainstorm some potential extensions of this work, or maybe even some entirely new research directions that it could inspire.
Yeah, let's do that. So the authors themselves mention the desire to incorporate more data modalities beyond just gene expression and text. What if we could take this core idea of cross-modal learning and really run with it?
I like where you're going with this. What if we could build a model that understands not only which genes are active in a cell, but also how accessible the DNA is, what proteins are present on the cell surface, maybe even spatial information about where the cell is located within a tissue.
And then imagine linking all of that rich, multi-layered information to a constantly updated knowledge base of all the biological literature out there, including scientific papers, textbooks, databases, the whole shebang.
That would be amazing. You'd be like moving from a flat, two-dimensional sketch of a cell to a fully immersive three-dimensional model with a whole library of information attached to every component.
And if we had that kind of hyperdimensional understanding of a cell, could we then start to think about not just analyzing existing cells, but actually designing new ones from scratch?
That's a fascinating idea.
I mean, the current paper shows that they can already generate pseudocells based on text descriptions. What if we could take that a step further and specify the desired characteristics of a cell, maybe an immune cell that's super effective at targeting a specific type of cancer, and then have this super smart multimodal model generate a blueprint for that cell, telling us exactly what kind of gene expression profile, DNA accessibility landscape, protein markers, and even spatial location would be needed to make that cell a reality.
That sounds almost like science fiction, but given how fast this field is moving, it might not be that far-fetched.
Yeah, I mean, SCM-GPT is a huge step towards being able to speak the language of cells in a much more comprehensive way. If we could scale this up to include all those other modalities and develop even more advanced generative techniques, maybe drawing inspiration from the recent successes we've seen in generating images and molecules using AI, we could potentially enter an era of truly designing biology at the cellular level.
That's a mind-blowing thought. Imagine being able to engineer cells with specific functionalities to treat diseases, create new biomaterials, or even, build entirely new forms of life.
And think about the implications for understanding and treating diseases. If we could create highly accurate digital twins of diseased cells, incorporating all those different layers of information, we could potentially run countless virtual experiments to understand why those cells are behaving the way they are and test different treatment strategies without ever having to step foot in a wet lab.
Yeah, that would be incredibly powerful. Of course, we need to be very careful about the ethical implications of this kind of technology, but the potential benefits for human health are immense.
Absolutely. And SCM-GPT feels like a really important stepping stone on the path towards that future.
It definitely does. I'm really excited to see where this research leads and what kind of amazing new discoveries it will enable in the years to come.
Well, on that note, thanks for joining us for another deep dive. We hope you found this exploration of SCM-GPT as fascinating as we did. And until next time, keep thinking deep.